hope you all are revising. Like, we are giving you assignments like on a daily basis for classes, right? Whenever we have a class, we give you assignments. So the purpose of that is to bring you in a habit of revising what you have been learning on a daily basis. Otherwise, if you are thinking I'll just note everything down in my notebook and just before the exams I'll open it and revise all of it, it's not going to work. It's difficult actually. I'll not say it never works, but it's very, very difficult to pull off with the last minute revisions only. You understand? Can you see the screen that I'm sharing with you, all of you?
Yes, sir.
Okay, so what did we study in the last class? Just a brief summary. You understood just the basic lineup of Calvin cycle, C3, C4 pathway. Anyone would like to just summarize just the basic crux in 2 sentences?
So, actually, can you explain the Kelvin cycle once again? At that time, I was not there. I got disconnected.
Yes, I will. But before that, others, Abhinav, would you like to just give a brief summary, just 2-3 lines about the last class? What was the take home message from the last class? What did you learn? Did you learn anything at all? Yes, I'm sure you must have learned something. You there, Abhinav? Abhinav, are you petrified, scared? Where is he? Okay, so by the time Abhinav is gathering some courage, so let me just revise for you a brief recap of the Kelvin cycle. That's what you want, Aisha, right?
Yes, sir.
Okay. So Kelvin cycle, basically, you can see the screen, right? And my cursor as well.
Yes, sir.
Yeah. So basically, Kelvin cycle is the pathway, the basic pathway through which we start fixing the CO2 from atmosphere. The plants start fixing CO2 from the atmosphere. Okay. Now, for how I started the class was I asked you to imagine the kind of molecule. If CO2 is to be fixed from the atmosphere, it has to be taken in by the plant, and some molecule must be accepting that CO2, right? So there should be a CO2 acceptor, correct? And since CO2 is a single carbon molecule, it has one carbon. And we know that in a C3 Kelvin cycle or a C3 cycle, the first stable product is a 3 carbon molecule, which is this 3 PGA. Is it clear? So naturally,
Yes, sir.
people thought that the accepting molecule, the CO2 acceptor, must be a 2 carbon molecule, which was not the case. Okay. So actually it is RUBP, right? It's ribulose bisphosphate. It is a 5 carbon molecule. So it accepts one CO2 every cycle and tries to fix it. So it itself is a 5 carbon molecule. It accepts one CO2 every cycle and makes 3 PGA, which is a 3 carbon molecule. So this 3 PGA, 3 phosphoglycerate, is the first stable product of this Kelvin cycle, where we have taken CO2 in a gaseous form and have successfully fixed it in a 3 carbon molecule. Is it clear?
Yes, sir.
From this step onwards, the next step is, this is called carboxylation step. Okay, where we are the carboxylation of RUBP is happening. RUBP is accepting a CO2. This 3 PGA, after utilizing energy from the ATP, so ATP comes and ADP goes out, and the phosphate gets added to this 3 PGA. It becomes 1,3 bisphosphoglycerate. So it was 3 phosphoglycerate. Now it became 1,3. Now there are 2 phosphates, so it becomes 1,3 bisphosphoglycerate. Is it clear? And this 1,3 bisphosphoglycerate now is reduced. So the next step is reduction. So you have to reduce this 1,3 bisphosphoglycerate by adding hydrogen from NADPH So NADPH comes, gives hydrogen and become NADP. This plus should not be here. NADP, and it becomes glyceraldehyde 3-phosphate. Now this molecule, glyceraldehyde 3-phosphate, is the output, is the final product that we get from Calvin cycle. So if you balance this equation, every 3 cycles, when 3 cycles are completed, one glyceraldehyde 3-phosphate comes out of this cycle, and rest of glyceraldehyde 3-phosphate goes back into the cycle to regenerate RUBP, because this RUBP, ribulose bisphosphate, has to be there so that this cycle can continue, so that over and over again you can accept CO2 and keep fixing CO2. Is it clear? So there are majorly 3 phases: carboxylation, reduction, and regeneration in a Calvin cycle, which is also a C3 cycle. C3, why C3? Because the first stable product of this cycle of carbon fixation is a 3-carbon molecule. Now there are many questions associated with this cycle itself. How many ATPs are utilized per cycle? How many ATPs are utilized to form one molecule of glucose? So to understand all that, you have to understand this cycle, that it has to go 6 times to fix 6 carbons so that one molecule of glucose can be made. Okay. Is it clear?
Yes, sir.
Yeah. So whatever happens in one cycle, you multiply it with 3. Here I have balanced this cycle, like the balance the reactions of this cycle, and it is for 3 cycles itself. So 3 RUBPs taking on 3 CO2, and then what happens? Now this this G3P, glyceraldehyde 3-phosphate, is also called a triose phosphate because there is a 3-carbon molecule, and this further goes on to make glucose or sucrose, starch. Is it clear?
Yes, sir.
Yes, I should write it here. Sucrose, starch is formed from this one. Now, I also, so is it clear? You just wanted to ask the C3 Kelvin cycle? Is it clear?
Yes, sir. Thank you so much.
Okay, fine. Then we also studied the C4 cycle, and we figured out that C4 plants or C4 pathway is evolved because these plants can effectively, more efficiently do this photosynthesis in higher temperature regions, in arid environment. Okay. And to carry out C4 pathway, you need to have a different special anatomy, which is known as Kranz anatomy. I told you about this Kranz anatomy. It involves mesophyll cells in the outer layer and bundle sheath cells in an inner layer. So the C4 plants have a different anatomy than C3 plants. Is it clear? And that's how they can pull off this difference, this different pathway, which is more efficient because, and it's called C4 because the first stable product of carbon fixation is not a 3-carbon molecule, it's a 4-carbon molecule, a C4 acid, which is called oxaloacetic acid, which stays in the cell as oxaloacetate. And this gets, oxaloacetate gets converted into other different types of C4 acids called malate, aspartate, etc. And this is all happening in the mesophyll cell. So technically, if you think, so everyone, this is just a general thing, if you understand something that I forgot to tell yesterday. If you think, there are 2 different kinds of mesophyll, 2 different kinds of photosynthetic cell in C4 plant, correct? Mesophyll and bundle sheath cells. This I told you yesterday.
Yes.
Out of which the mesophyll cell, one type of photosynthetic cell, does not have rubisco, right? No rubisco
Yes.
enzyme is present in mesophyll cell. Is it clear? We have Mariam also joining in. Hello, Mariam. So the mesophyll cell has no rubisco. So if this comes as a question, you should understand the rubisco is only present in the Calvin cycle, which happens in the bundle sheath cells. So in a C4 pathway, this C4 acid is first this HCO3 is first carboxylated through PEP, and upon carboxylation, it becomes OAA, oxaloacetic acid. Then it's transported and gets decarboxylated to remove CO2 back. Now, if you remove CO2 from a C4 acid, one carbon is removed, so a C3 acid is left, which is a pyruvate. Now, this CO2 then goes into the Kelvin cycle, and everything that I told you above in Kelvin cycle is also happening in a C4 plant. So Kelvin cycle is common for both C3 and C4 plant. The only difference is that this the presence of PEP and PEP carboxylase for the carboxylation of PEP along with HCO3 and it forming a OAA is not there in C3. And the anatomy is also different. It does not have two type of photosynthetic cell. It has only one type of photosynthetic cell, whereas C4 has two type of photosynthetic cells. Is it clear, everyone? All is good till now?
Yes, sir.
Okay. Now in today's class, we understood that what is the advantage of, and I also told you about photorespiration. Everyone is clear with photorespiration? Photorespiration, RUBP plus CO2 in the presence of, so photorespiration is basically, it's not present in C4 plants. That is one benefit. You understand, everyone? Is it clear or not? Should I go over photorespiration once more, or is it clear?
I was not there.
It's not clear?
No.
You have written photorespiration in last class?
No, sir.
Okay. So let's begin with, let's get into some details of photorespiration. Write down photorespiration. Let's spend 5-10 minutes here. So one thing is respiration, correct?
Yes, sir.
Cellular respiration. What happens in cellular respiration? Respiration I've also taught you in, we have talked about cellular respiration multiple times, like right from the first chapter onwards. We know it's a characteristic property of life, metabolism, cellular respiration, right? What happens in cellular respiration? Yes.
It's the process by which organisms combine oxygen with food molecules
We are not
to
combining.
produce chemical energy.
Yeah,
No, something like that only. Like, no, organisms use oxygen to break down food molecules to get chemical energy for the functions of cell or ATP.
Yeah, we don't break all the food molecules. We are not breaking lipids, vitamins, proteins. What are we breaking? It's specifically glucose. Our cells break down glucose in the presence of oxygen. So you should not say breaking down food molecules. There can be multiple thousands of different molecules in food. But ultimately, the energy source or the fuel comes from the sugar, glucose in our body. Okay. So that glucose is broken down in the presence of oxygen, more like combustion. Remember, we talked about this, that what happens during when something is burning, it's similar like when we are doing respiration in our cells, correct? When we are breaking down glucose in the presence of oxygen, it's a kind of a combustion reaction itself, right? The only trick is that we are able to do it at a very low temperature in a controlled bio system. Otherwise, if you burn a biscuit versus you digest and utilize the glucose of that biscuit, it's the same thing. You need oxygen for burning the biscuit and the temperature will be very high. You understand? So basically we are doing the same thing in our body, in each and every cell of our body, in a controlled way. We are breaking down glucose, utilizing through oxidation of it. And when we are breaking down bonds, we are getting more and more energy out of it. Because bonds are nothing but conserved energy, right? When you break it, a lot of energy comes out, correct?
Yes, sir.
Yeah, okay. So but photorespiration is something different. It's specific to plants and photosynthesizing organisms. What happens, as the term says, the word photo means light. Photo means light. And respiration is respiration. So this is a light dependent. So write down photorespiration creates an important difference, okay, between C3 and C4 plants, whatever we have studied till now, C3, C4 cycle. So C3 and C4 plants, photorespiration makes a big difference. Photorespiration happens in C3 plants. Okay. Happens in C3 plants. Yesterday I defined photorespiration. Can someone just repeat it for me? Photorespiration, please. Can anyone repeat that definition?
Yes, sir. A process of light-dependent uptake of oxygen, which leads and giving out of carbon dioxide.
Yes. That's it?
And then, yeah, yes, sir. And then there's another point, the gaseous exchange.
Yes, right. So basically, what we are doing is we are taking in oxygen, but in a light dependent manner, correct? Okay.
Yes.
Yeah. So in C3 plants, it happens in C3 plants, but it does not happen in C4 plants, or like there are some reports that it's negligible or almost absent. So you will find words like less, absent, negligible, but it happens in C3 plants majorly. C4 plants can do without photorespiration, and that's why they are well adapted to hot and humid, hot and dry conditions. So write down one more thing. In C3 plants, I also told you yesterday that why photorespiration is not good for photosynthesis. Because if photorespiration is happening along with photosynthesis, remember I told you that the enzyme Rubisco, which catalyzes the reaction of binding of CO2 with RUBP, the first reaction. Please pay attention and keep saying something. This is RUBP, ribulose bisphosphate, and it has to bind with CO2 to form the first stable product of Calvin cycle, which is 3-phosphoglycerate, phosphoglyceric acid, PGA, correct? This is the first reaction.
Yes, sir.
And I told you that it's catalyzed by this enzyme, Rubisco. And this is the most abundant enzyme. Now, Rubisco can also do carboxylation. It can also add oxygen to RUBP if oxygen is available in higher quantity. Remember, I told you it's a dual enzyme. It is capable of doing both carboxylation and oxidation of RUBP. Clear, everyone?
Yes, sir.
Now, why photorespiration is a bad thing for photosynthesis? Because if through photorespiration, lots and lots of oxygen can come in. Remember, it is the uptake of oxygen in a light dependent manner, correct?
Yes, sir.
So if more oxygen comes in, then Rubisco, it will compete with CO2 for Rubisco to oxidize RUBP. If RUBP gets carboxylated, then only we can form this first product of Calvin cycle, which can further go on to form food, glucose, starch. But if it gets oxidized, this will not happen, correct?
Yes, sir.
So the more photorespiration happens, the less photosynthesis happens. Is it clear, everyone?
Yes, sir.
So that's the problem. And if C4 plants can get rid of photorespiration or can just just nullify it or get rid of it completely, then obviously at higher temperatures in a high light intensity manner, they can still be able to do more photosynthesis because oxygen will not come in through photorespiration to compete with CO2. Correct?
Yes, sir.
So write down about photorespiration. In C3 plants, some oxygen O2, in C3 plants, some O2 can bind to Rubisco, leading to some O2 can bind to Rubisco, leading to decrease in CO2 fixation. Decrease in CO2 fixation. Is it clear?
Yes, sir.
So that's why C3 plants cannot do well in high light intensity area, because if there is more and more light, photorespiration will happen. Okay. And in C3 plants, photorespiration does happen. In C4 plants, because the first thing that is, so if there is lots and lots of sunlight, what is present in the outer side which will encounter all that sunlight? Mesophyll cells. Correct? Correct, everyone?
Yes, sir.
And do we have
Yes,
rubisco in mesophyll cells? No. We do not have rubisco in mesophyll cells. So photorespiration cannot happen, because if oxygen comes in, even if oxygen comes in, it will not compete with CO2 for its fixation, because now the fixation of CO2 is happening through PEP carboxylase and not rubisco. And the first stable product is C4OAA. And only when it is getting transported into the bundle sheath cell, which is on the inner side, in the inner layers, there we are again taking back CO2 and giving it to the Kelvin cycle. So no, we are getting rid of the competition with oxygen, correct?
Yes, sir.
yes, sir.
Everything is clear till now. This is the photorespiration is clear.
Yes, sir.
Yes. So it competes. Now write down, in C4 plants, photorespiration is absent. Let's consider it absent for now. In C4 plants, photorespiration is absent. And they have a mechanism, which is this mechanism, using PEP through PEP carboxylase. And they have a mechanism that increases the concentration of carbon dioxide. They have a mechanism that increases the concentration of carbon dioxide that is available to Kelvin cycle. That is available to Kelvin cycle. In the bracket, write down rubisco. So basically, they make sure that only CO2 goes to rubisco and not oxygen. Correct?
Yes, sir.
Yes. Very good. Now, is there any doubt in photorespiration? Please ask if you have any doubt still. You know, photorespiration is still under research. It's a lot of research is going on that. And write down another point. Unlike respiration, there is no energy produced in photorespiration. So why we respire? Why do our cells do respiration? Because we have to make energy from the food, correct? Photorespiration does not even make any energy, but still happens. So the detailed molecular mechanism of it is still not completely known. There are a lot of research still going on, but you have to understand all these points. Is everything clear till now?
Yes, sir.
Yes, sir.
Okay. Now, let's come to certain factors, because this is an important point. Through photorespiration, I got. Okay. Sometimes the pen does. Okay. Factors affecting photosynthesis. All these factors we have already talked about. So every ingredient is basically the factor that can affect photosynthesis, correct? Everything that is involved in the process can only affect it, right? So what factors can you think that can affect photosynthesis? Let's start with the very simple one, which is light. Let's start with the photo itself. Do you think light can affect photosynthesis?
Yes.
How? Light is required for photosynthesis.
High intensity, low intensity.
Yes, but light is required for photosynthesis, isn't it?
Yes, sir.
So in a way, if I tell you that the more the light is there, the better the photosynthesis will happen. Is it correct?
No, sir.
No.
Why? Why, Abhinav?
Sir, because photosynthesis is a process which is completely dependent on enzymes. So it may happen that the temperature may increase when the light intensity is increased. So the enzymes may not get activated.
But we have not yet come to temperature. Light all the time does not mean that there'll be a so much significant increase in temperature, or will it always?
Not always, sir, but it can be.
Mhm. Then how do you think? So let's not merge light and temperature together. Okay. Let's say that if I, because I'll tell you why I'm not merging temperature and light, because when you sit in sun, you think that, after 4-5 minutes you are feeling very hot because the sun gives you constant heat, right? That's the radiation. But heat is also a form of radiation. Sometimes you can still get heat without any light, and it happens all the time, right?
So it's used in hospitals, right? Most of the times I have seen
Yes.
that
So
yellow
oil
light
heaters,
and all.
the oil heaters, they do not give
Yeah.
out light energy, but they give out constant heat energy. So,
Yes, yeah.
and there is one more reason why I'm not merging temperature and light here, which I'll come to, but let Abhinav finish his answer. Yes, Abhinav. If you do not take temperature factor into consideration, can you just say how simply light is not good if it keeps increasing for the photosynthetic process? Because normally we think it's light dependent, so more the light, better the photosynthesis. But why? Abhinav, you there?
Yes, sir.
Yeah.
I'm thinking, sir.
Okay, let me lead you towards that answer. What is light? Why is light being trapped at all for photosynthetic process? What is light giving to the process? It's not giving heat to the process, right? We are not.
Energy.
Yes, it's giving energy to excite the electrons in the photosystem. LHC, light
Yes, sir.
harvesting complex. Light harvesting complex. And what is a part of the reaction center of that light harvesting complex is chlorophyll A in the center, one single molecule, and other accessory pigments around, correct?
Yes, sir.
And when light falls in it, it has a particular energy which excites the electrons. If light intensity keeps on increasing, very high and high and high light intensity, then what will happen? There is always a chance of what?
Photooxidation.
Of what?
Chlorophyll.
Exactly. So there is a chance that chlorophyll A itself can get, broken up. There can be a breakdown of chlorophyll, a like light dependent breakdown at higher intensities. It's like our eyes. Let me give you an analogy. Can we see very well in dim light?
No.
Can we see very well in very high light?
No.
Exactly. Why? Because there's a range in which our photoreceptor cells in the retina can get excited optimally and do their best work. So very high intensity of flashes to the eye can also lead to breakdown, photo oxidation of light dependent breakdown of those receptors. Is it clear, Abhinav?
Yes, sir.
Yes, sir.
Yeah. So write down light. And if I draw a graph, it's very, this is a very classical graph where on the x-axis we have light intensity, and the y-axis we have rate of photosynthesis. So how do you think the graph will look like? Starting from the 0, initially it's linear, like when light intensity increases, rate of photosynthesis increases, okay? But after some time it saturates and it stays saturated. Is it clear?
Yes, sir.
Because as the light intensity increases, after one point of time, just like how our eyes respond, after one point of time, if you, it's just like you can do the same experiment if anyone has a digital camera. If any one of you has a DSLR or a digital camera, or in your mobile phones, yes, of course, everyone has a digital camera now. Go to the settings, pro mode, and go to the brightness while you are capturing it. So if you keep increasing the brightness, the field will start getting more and more and more saturated. After one point of time, there'll be nothing but just a blank white screen. The camera is not capturing anything. Or in other words, I should say, it's capturing everything, and it's not able to figure out what image or what figure is what. It happens, right? Same is with the eyes. As the room starts getting brighter and brighter, we can see things very clearly, which is this linear phase. Dim light, less light intensity, we cannot see. We can see clearly. After one point of time, all our photoreceptors get saturated and we cannot see in a very high intense light. Same happens with, so how photosynthesis is affected by light, write down. At low light intensities, there is a linear relationship. At low light intensities, there is a linear relationship between the incident light and the rate of photosynthesis. Between the incident light and the rate of photosynthesis. At higher intensities, rate of photosynthesis saturates. At higher intensities, rate of photosynthesis saturates. And beyond a high threshold, it can cause breakdown of chlorophyll. And beyond a high threshold, it can cause breakdown of chlorophyll, leading to decreased photosynthesis or decreased rate of photosynthesis. So if you keep increasing light intensity, after one point of time, you will see that the graph goes like this. Correct or not? If I keep increasing the light intensity beyond like something, a very high threshold, we will see this, because at this point of time, from this point onwards, chlorophyll breakdown started, right?
Yes, sir.
And what is this actually? You know, you will be very surprised to know that all the light on a bright sunny day when there are no cloud cover and the sunlight can directly come to surface of the earth, this saturation reaches at just 10% of the total sunlight. Is it clear?
Yes, sir.
So this is where I was not taking temperature factor into consideration, Abhinav, because 10% of the total sunlight will not cause that significant increase in temperature, correct, Abhinav?
Yes.
So basically, photosynthesis is saturating way before temperature factor can come into picture because of sunlight, right, Abhinav?
Yes, sir.
Yes. And this 10% also tells us that if 10% can saturate the photosynthesis, that then even on a cloudy day, light is not a problem for plants, right? At least 10-20% must be still coming through the clouds. It's still a day where we can get some light, right? Even if there is a cloud cover, you can see things, which means light is coming through it. Is it correct or not?
Yes, sir.
It means it's enough for plants, which also means that in a very dense forest also, it's not a problem for plants till those plants are small grasses or shrubs at the very surface where no light can reach. then it becomes a problem. Otherwise, every plant can do good with the sun and its sunlight in all sorts of weather. Is it correct? So this is the effect of light. Everyone's clear? It increases, it has a linear relationship till some point, then it saturates. And if you go beyond a certain high threshold, like 90%, 100% of the sunlight, chlorophyll starts breaking down and photosynthetic rate again will come down. Cool? Any doubts?
Yes, sir.
No, sir.
No. Okay, now let's talk about temperature, which Abhinav was talking about. Okay, let's go to temperature separately. Yes, Abhinav, anything you want to say about temperature? Now,
Anything more or what I said?
yes, so what you said like makes sense here, right? You can just
Yes.
So your answer was for temperature, which you gave it in light. So if in boards they ask you about light, stick to light, don't go to temperature. Because we associate it as humans most of the time, because more sunlight is a hot day, but it's not true for like same for plants. So yes, about temperature, you were saying that because these reactions, okay, there are two reactions, light reaction and dark reactions conventionally in photosynthesis. Which will be more affected by temperature, both equally or no? Abhinav, because you answered about temperature.
Light reactions, sir.
Why light reactions?
Sir, because the other one is said to be not dependent on light, so it doesn't use light only.
Yeah, but what happens in the light reactions? Can you just give me a... See, it's interesting. It's interesting how you are relating it with like dark and light reactions. That's an interesting thought process. Let's see if it's true or not. So what happens in light reactions, Abhinav?
So the light is absorbed and it is used for like in the process of photosynthesis.
Yes. So which means the only thing that can be problematic in a light reaction is if high intensity of light comes, it can break down the core, reaction core, which is chlorophyll. You know, is there any enzyme which is involved in light reaction? It's just passing of electrons, right? And finally it's going into NADP plus to become NADPH.
Yes.
But in the dark reactions, which we say dark reactions, basically we also say it carbonic reactions or biosynthetic reaction. There we have enzymes like rubisco playing an important part, like PEP carboxylase playing an important part, right?
So it affects the dark reaction.
Exactly. You know, I didn't give you the right answer. I asked you to go toward, and now you understand why it affects dark reaction and not the light reaction.
Yes, sir.
Because your reasoning was correct. You gave that reasoning right away for light only. You said that there are enzymes which can get affected at higher temperature because every enzyme has an optimal range in which it works the best. Correct. Good reasoning. But you have to understand where to apply that reasoning. So write down temperature. The dark reactions are more temperature controlled. The dark reactions are more temperature controlled as they are enzyme dependent. Now write down light reactions are affected by temperature to a lesser extent. It's not like Abhinav is not completely wrong in saying that light reactions will also get affected. It's not like whatever with the temperature, light reaction will just keep happening, because ultimately temperature will also, if there is light bringing in energy, heat will also bring in energy. Correct or not? Ultimately we have to excite an electron. You can excite it through radiation, which can be heat or light or sound, like any radiation, any not sound, but like electromagnetic, infrared, ultraviolet, anything which can impart energy to the electron. Correct? And heat is also a radiation. So it's not like that light reactions will not get affected, but generally they do not get affected to that extent. Like it's very, very minimal, the effect. So write down, light reactions are affected to a much lesser extent by temperature. So at high temperatures, photosynthesis decreases. If temperature is high, then photosynthesis is high or low? Low, correct? Say yes or no?
Yes, sir.
In general. But the important, interesting thing here is that temperature have different effects. Write down, temperature affect C3 and C4 plants differently. Temperature affect C3 and C4 plant differently. Can anyone tell me? Because by now you already know it. So who will tell me?
C4 needs more higher temperature.
It does not needs or wants.
It works.
Yeah, it can respond even well in a high temperature. So write down, C4 plants can respond to higher temperatures. C4 plants can respond to higher temperatures and can still maintain high rates of photosynthesis. So C4 plants, for C4 plants, this thing does not happen, correct? At higher temperature, C4 plants do not show lower photosynthesis. For C4 plants, at high temperature, they can still show high photosynthesis. Clear, Abhinav? This is why they can survive in hot and arid environment. This is why how they are adapted. They can get rid of photorespiration. Okay. And write down, whereas for C3 plants, higher temperature leads to reduced photosynthesis. For C3 plants, so this is basically correct for C3 plants. Higher temperatures leads to reduced photosynthesis. And it's not only about C3 and C4 segregation. Temperature can also affect on the basis of habitats, right? Different habitats have different types of plants. Now, in a habitat, there can be both C3 and C4 plants, but living in one habitat. Do you understand? So plants which are basically in the tropical region, like we are in a tropical region. So write down, plants in a tropical region have higher temperature optimum. This can also be asked. In a tropical region, tropical plants have higher temperature optimum. What does it mean, temperature optimum? In which they can still work, right? Whereas temperate plants, temperate region plants or temperate plants have low temperature optimum because they are adapted. In evolution, they are adapted to that. Sorry, यहाँ पे M नहीं होगा, optimum. Is it clear, everyone? The effect of temperature?
Yes,
Yes, sir.
So the effect of temperature can be, three reasons. One, higher temperatures anyway affect enzymes. So enzymatic activity decreases, and since the dark reactions are more dependent on enzymes, it will be affected. Second thing, In C3 and C4 plants, the effect is different. C3 plants at very high temperatures, their photosynthetic rate will start decreasing. But for C4 plants at high temperature, they can still keep maintaining high photosynthetic rates. But for C4 plants also, even beyond a temperature where their enzyme starts degrading, photosynthesis will also degrade, and so will all other physiological processes, right? Third way it can affect is through habitat. So some plants which are always, they grow in tropical, gets adapted to tropical. Okay, whereas plants which grows in temperate, whether they are C3 or C4, they gets adapted to temperate accordingly. So the effect of temperature is not same. It depends on certain factors, cofactors again, like certain other sub factors. Is it clear, everyone?
Yes, sir.
Yes, sir.
Right. Now let's get to other factors. What could be the third factor? Light and temperature is done. Let's talk about carbon dioxide itself, CO2. Can CO2 affect photosynthesis or no? Of course. CO2 is the major ingredient of photosynthesis, so it can affect the photosynthesis the most, correct? If you are making an egg curry and you do not have egg, it's going to affect your recipe, correct?
Yes, sir.
Yes. So there is CO2. How can CO2 affect us? Write down, it's the major limiting factor for photosynthesis. This was also asked. CO2 is the major, the most important limiting factor. We should have done it the first, but okay. Major limiting factor. Now, if there is more CO2, if I'm asking, because CO2 is required for photosynthesis, correct? Just like light, we need light for photosynthesis and we need CO2 as raw material for photosynthesis. Now, if I ask you, what do you think is the case of CO2? If CO2 keeps on increasing in the atmosphere, will photosynthesis rate will also keep increasing?
Yes.
Yes. So the more CO2 in the atmosphere, the better the plants will work or do photosynthesis, according to you, correct? Mariam?
Maybe.
Maybe. Okay. Abhinav, what do you think? Abhinav is
But
very
they need oxygen also for survival, no?
It needs oxygen. So oxygen is like, let's talk about our atmosphere now. So we have around 21% oxygen. And what is the concentration of CO2 in atmosphere?
Less than 1%.
What? Less than 1% could be 0.9, 0.8, 0.7,
But that I don't remember.
Okay, it's very, very less. It's 0.03 to 0.04. When we look at these figures, we are thinking why this whole world is talking about CO2 rise and global warming. CO2 is 0.03. Like, what will do the worst? Like 1% tak pahuncha denge, kya hoga? What's the problem? Why are we worried? We are worried because CO2 is a greenhouse gas. You know that?
Yes, sir.
And what is a greenhouse gas? What is a greenhouse gas? What is a greenhouse? A greenhouse which is painted green. You know what is a greenhouse, right?
Yes, sir.
Yes. What is a greenhouse, Maria?
Plants in a glass type of.
Yeah, you'll see in colder countries like in Europe, they always grow their tomatoes in a house which is made up of glass. Basically what it does, it traps, because there is very less sunlight, very less light energy or heat energy, and it's also required for enzymatic activities, and it's also required for CO2 concentration. So greenhouse basically traps the heat energy and does not let it go outside. So it's like trapping of heat. Now this CO2 acts like a greenhouse on the globe. Even if its concentration increases from like 0.04 to 0.05, it is very insignificant in terms of mathematics, and like we'll say, okay, 0.04 it got to 0.05, what's the big deal? But the big deal is all these extra molecules of CO2 in the atmosphere will start shielding the atmosphere, because all the light that falls on the planet Earth's surface, most of it gets reflected back into the space. Correct?
Yes, sir.
And whatever stays in the atmosphere is used by plants, animals, living systems, and also heats up the air. So the mean average temperature of our planet Earth should be in a balance. It should be such that certain areas of our planet stays cold and certain areas stay hot and certain areas stay moderate. It can never be the same everywhere, because if it is same everywhere, then what is the mean average temperature of Earth? Anyone knows? Mean global temperature? Around 15 degrees, 13, you understand? But if you go to Arctic and Antarctic, lots and lots of glaciers are there, which clearly means the temperature there is sub-zero. And if you go to Africa, clearly the temperature there is over 40, 45 in summers. And that balance has to be made. That balance can be made if lots and lots of greenhouse gases comes into picture. They will increase this average, mean average temperature of Earth, which will start melting the ice. An ice starts melting the moment it comes even 0.1 degrees above the freezing temperature. There will be molecules that for them, this energy will be enough to convert from solid phase to liquid phase. Do you understand what I'm trying to say?
Yes.
Yes, sir.
So at 1 degree Celsius, there is water, very chilled, but still water, right? It does not freezes. Have you ever seen any ice freezing at 3 or 4 degrees Celsius? To make something ice, you have to go below zero, right? Sub-zero
Yes, sir.
temperatures can only freeze
Yes,
water.
sir.
And even if it goes slightly above zero, it will start melting and the water will be chilled, but it will be water, liquid water. And that's the issue that's happening with global warming. All this ice, which is all this water, which is trapped in the form of glaciers and ice, even if it start dripping 1 drop per day, it's melting. And all that water, if all that ice becomes water, first of all, it will flood all the oceans. So all the cities which are at the edge, like all the beach cities, will just submerge. Secondly, this ice sheet, both on the north and the south, acts as a reflector. So it's a feedback. Do you understand? More light coming in and staying in the atmosphere will melt down more glaciers, which will cause more light to stay in the atmosphere because it will not be reflected back. So it's not just one thing that's increasing. It's just a domino effect. Just like CO2 increasing from 0.04 to 0.06 is the first domino that you topple. And after that, a series of dominoes gets toppled and global warming escalates. Do you understand?
Yes, sir.
That's the problem. That's why we are worried about CO2. Now, coming back to the actual question. If there is more CO2, will it lead to more photosynthesis? To a certain limit, yes. Just like the relationship with light. So if it goes till 0.05, something around it, it's fine. When you increase CO2, more CO2 gets fixed because more of it is available. But after a certain point, it becomes damaging to the photo systems in a long run. Do you understand that? Okay. So in a longer period, more CO2 is not good for photosynthesis. So that's why we are worried about that if our CO2 levels goes beyond a level where first it is heating up the environment, second it is also causing damage to the only system present on the earth which can fix it, which is the photosystems in the photosynthetic pathway. So we are losing on both the fronts. So after certain period of time, there will be no efficient photosynthesis which can fix CO2, and there will be nothing which can control it. So it will be a domino effect and it will topple so many dominoes that we will not be able to reverse it back. Clear, everyone?
Yes, sir.
Yes. Now again, just like temperature, C3 and C4 plants have different tolerance, or they show different responses to CO2 concentration. But first write down. CO2 is a major limiting factor, and the concentration of CO2 in atmosphere is extremely low, is very low to begin with. Is very low to begin with. Which is 0.03 to 0.04%. A slight increase in this concentration, will lead to higher photosynthesis. In brackets, write down higher CO2 fixation. But beyond a certain level, which is write down 0.055, is that, if I'm not wrong, that level threshold is 0.05 to 2 decimals position. 0.55. Beyond this level, CO2 has a damaging effect in a long run. Is it clear?
Yes, sir.
Yes. Now write down the effects of CO2 on photosynthesis is different in C3 and C4 plants. The effect of CO2 concentration on photosynthesis is different for C3 and C4 plants. And this effect is associated with the light conditions, the availability of light. And this effect is associated with light conditions. Like the effect of CO2 is associated with the light conditions. So at low light conditions, write down at low light conditions. So when the light condition is low light condition, both C3 and C4 does not respond to high CO2 levels. Is it making sense to you? Can you pin the wires? Because when light is low, what is happening?
Low chemical energy.
Yeah, the system is not saturated.
Yeah.
Yes. And when it is not saturated, it does not need higher CO2s. Whatever is the amount of CO2 in the atmosphere, it's fine for it. It's working well in that. So it does not respond to high CO2 levels. Making sense?
Yes, sir.
But at high light intensities, write down at high light intensities, or high light intensity conditions, both C3 and C4 shows increased rate of photosynthesis. Increased rate of photosynthesis. Increased rate of photosynthesis. If there is at photosynthesis at high CO2 levels. Okay. Because what's happening at high light intensities? Basically, it should saturate, right? Because of intensity, the rate of photosynthesis is saturating. Are you with me, everyone?
Yes, sir.
Yes, sir.
So if at high light intensities, photosynthesis is not happening, then you have to push the photosynthesis. And you can push it by giving more and more CO2 for fixation, so that O2 does not competes with Rubisco, but there is only CO2. So at higher light intensities, high CO2 can work for photosynthesis, seems to be working for photosynthesis. Is it correct? And that is, this rate is also different. C3 saturates, write down, C3 saturates at C3 saturates at 450 micro 450, what happened to the pen? $450 microliter per liter of CO2 concentration. Okay. And C4 saturates, I'm talking about highlight condition only. And C4 saturates at somewhere around 350 microliter per liter. 350, 360. Let's make it 360 of CO2 concentration. Now it's clear that even at high light intensities, which of the two systems is showing more photosynthesis at higher CO2 concentration? Or in other words, if I ask the question like this, which is asked in boards like this, which system responds to higher CO2 levels better?
C3.
C3. Okay. Yes. C3. Because of which there are many C3 plants that are used for greenhouse purposes. Can you go and find out which are these plants which are used at, which are grown at enriched CO2 levels in a greenhouse? So excess CO2 is pumped into that greenhouse and they are grown at higher CO2 levels because they show more productivity, because they make more and more starch, they make more and more fruits. So anyone can guess, or will you find which are these plants? Anyone knows, you can tell me. If you don't know, it can be asked in entrance. So will you find or should I tell you? Go and find, okay? This is the assignment. So which C3 plants are used at enriched CO2 levels for better growth, better production, and better photosynthesis? Is it clear?
Yes, sir.
Okay. So we talked about CO2, we talked about temperature, we talked about light. What other factors could affect photosynthesis? Is there any other factor you can think of?
Water.
Water. Okay, tell me, how can water affect photosynthesis, Mariam? Because you are saying it, let's just do it. Which is this third, fourth factor, na? Yes, let's take water. Okay, how do you think water can affect photosynthesis, Mariam?
Because if there is less water, the stress, plant stress, something.
Forget about plant stress. Where is water directly used in photosynthetic reaction? I told you.
Sir,
Yes, Abhinav.
sir, if water is less, then there'll be less breaking of H2O for the electrons used.
Yes. Remember, I told you water has to be that molecule that can ensure continuous supply to the photosystem too, because the electron in a Z scheme finally gets used up at the level of NADPH. We lose the electron. That's why it's a non-cyclic photophosphorylation. So the electron which starts its journey from LHC of photosystem 2 gets lost finally. It gets utilized. So so those plants which has both photosystem 2 and photosystem 1 needs a constant supply of electron, which could be water, which has to be water, correct, Mariam?
Yes, sir.
So that could be one place you can think that could get affected, right? But we are talking about electrons, okay? And since it makes sense to think like that, and I'll not burst your bubble, so you can write that for water as a factor, you can write that it's one of the reactants in the light reaction, okay? But still, even at like it does not affect the rate of photosynthesis directly, because plant has its way. Because if there is no water, still there is another way to make the photosystems work. Make it cyclic. Remove PS2, just keep PS1. So if you are a plant who's growing in a water deficient environment, why will you have PS2? Right, Abhinav?
Yes.
Or all those plants who have PS2 will just perish. So only plants which will survive is who have a cyclic photophosphorylation. So water is not required to make ATPs now. So you can still do the light reaction and still make ATPs without water. So that's why there is a counter mechanism that can compensate for it. So even if it is one of the reactants in light reaction, water does not seem to be affecting photosynthesis directly. Okay, so write down. Water being one of the reactants in the light reaction still don't affect photosynthesis directly. Just this one line. But still it can affect photosynthesis. Now let's come to what Mariam was trying to say. Yes, Mariam, let's hear from you.
Sir, the plant stresses if there is less water.
Then what happens? How
And
does
the
it affect?
stomata closes.
Yes. And then what happens?
Light.
Hey,
Transpirational pull won't happen.
then what?
Nothing.
Okay. No, you cannot just give up at the last moment. Mariam, you started right. So it can still affect photosynthesis indirectly. And let me push Mariam towards her answer. She started correctly that if there is water stress, if there is less water for plants, the plant have no other option than to close its stomata to reduce further loss of water through transpiration, right?
Yes, sir.
And because it's closing stomata, it does not want to do that. But when it's closing stomata, what is happening?
Carbon dioxide won't go.
Exactly. Won't come.
Won't come.
Yes, photosynthesis takes CO2 inside, na?
Yes, sir. Go,
Yes.
I mean in the leaves.
So if stomata is closed, then you manage to save water, but you also close the only doorway through which CO2 could come in for photosynthesis.
Yes, sir.
So that way it could affect photosynthesis, right, Mariam? Never
Yes.
give up on the last stage of your answer. Okay. Yes. So write down. It can affect photosynthesis through water stress condition. Through water stress condition, where the stomatas close, to prevent the loss of water, to prevent further loss of water, but also reduces the CO2 availability. Any other indirect way through which it can affect, Mariam, you can think of, or Abhinav, Reba? So it's not directly affecting the chemical reactions of photosynthesis. We have rehab also. Yes. Any other way through which it can affect? Water can affect.
The leaves won't be optimum at doing photosynthesis because
How? Because
they won't get food.
they won't get.
I mean, transportation of food won't take place.
Food, we have to make food. Photosynthesis is all about making food.
But then we have to take the food to other parts of the plant.
Yeah, that is not dependent on transpirational pull. Na? Transpirational pull only brings, it's like a lift, which only will take things from down to top. For getting food everywhere, we have phloem. But what happens when plants become dry? You're right about leaves. Again, never give up on your answer at the right last stage. So when plants dry up, their leaves wilt, right?
Yes, sir.
Have you seen leaves curling up and closing up in high temperatures? You will see it. Many plants, like especially in areas like Delhi and the northern belt of India, if someone has spent time here in summers are really very dry. We call it, there is heat strokes or loo, which blows throughout that north belt of India. Loo blows. Loo is it's like a very hot and dry air. Okay. And it causes plants to wilt their leaves, like the leaves try to close, which reduces the surface area on which light can fall and make photosynthesis happen. Right? This could be another way through which water stress can affect photosynthesis indirectly. Correct?
Yes, sir.
Yes, so you can write down. Water stress can also make leaves wilt, W-I-L-T. Wilt, which reduces the surface area available for photosynthesis. So the overall headline is that water does not directly affect the photosynthetic process, but can affect it indirectly. Any other factor that you think can affect photosynthesis? Oxygen? Can oxygen affect photosynthesis? People? I've just told you about that. Just say that answer. It's a one-line answer for oxygen. Can it affect photosynthesis?
Yes, respiration.
Photorespiration, not respiration.
Photorespiration.
If you write the word respiration, the teacher will give you a big 0. I told you that respiration and photorespiration are two different things. Respiration is good for the cell, it has to happen, but if there is more of oxygen in the photosystems, then photorespiration will start in C3 plants. So more oxygen will lead to reduced photosynthesis in C3 plants, right? So O2, high levels affect photosynthesis in C3 plants because of photorespiration. Okay, Abhinav?
Yes.
But will O2 do any effect in C4 plants? No. No effect in C4 plants, because in C4 plants there is no photorespiration. So C4 plants can do very well even if there is more oxygen inside the system. Clear, everyone? Is it clear or not?
Yes, sir.
Okay. So I think there is no other factor. So we have done I think we have completed everything that we needed to know about photosynthesis. Yes. So let's take a 3 minutes break. I need some hot water. I'm giving a 3-minute break. Just look at photosynthesis notes from top to bottom and let me know if there is any doubt. And then I'll come to glycolysis. Basically what we made in photosynthesis, now we have to break it down in the process of respiration to generate energy. So this is what we will be taking up next. So allow me a break of 3 minutes and go through photosynthesis notes. Okay. Okay, everyone.
Yes, sir.
Yeah, thank you. Hello. Am I audible and visible, people?
Yes, sir.
Yes, welcome back. Just take one more minute. Let me plug my laptop. Okay. Okay, so all clear in photosynthesis? Any doubts? Any doubt in photosynthesis, people? Say yes or no?
No,
No
sir.
doubts.
Okay, so if you have any doubts, please do ask. Okay. And if you have no doubts, so we will now begin with, so what we made, what plants synthesized during photosynthesis was finally starch or glucose, correct? Now this starch or the glucose has to be converted or has to be broken down during respiration. So let me start a new page, physiology, where we'll talk about respiration. Okay, can you see the thing? It's shared with you, right?
Yes, sir.
Yeah, all of you can see it, correct. Okay, so just continuing the discussion, we made glucose and we studied photosynthesis. Now we have to utilize it. But before going into the utilization of glucose, just tell me what do you understand? So you, by now everyone is clear that respiration, cellular respiration and photorespiration are two different things. Respiration is common for all living systems, correct? Remember chapter 1 when we studied that cellular respiration is a life process as well as a characteristic property of life, correct?
Yes, sir.
Because now respiration can be of different types, right? You must have known or you must have studied the term aerobic respiration and anaerobic respiration.
Yes, sir.
What is aerobic respiration? What is anaerobic respiration?
Respiration in presence of oxygen and anaerobic is absence of oxygen.
Okay. Now, absence of oxygen. Okay. Aerobic is, in a better scientific terminology to use is aerobic is oxygen dependent. Which means if there is no oxygen, aerobic respiration will cease to happen. It will not happen. It is dependent on oxygen. Whereas anaerobic is oxygen independent. Okay. And why I'm using this oxygen dependent and oxygen independent term? There are two types of anaerobes also. One is called facultative anaerobes. This is in microbiology. For bacteria, we use these terms. And one is called obligate anaerobe. Facultative and obligate. You know what does this mean? So anaerobic means where oxygen is not important. But if oxygen is present in the environment, let's say oxygen is present, but you are an anaerobic bacteria, will you not be able to do respiration? Tell me.
Sir, could you repeat?
If there is a bacteria which is an anaerobic bacteria, which means that its respiration is not oxygen dependent, okay, or oxygen is not required, or like you said, it happens in the absence of oxygen. So there are two different things. Oxygen independent process can still happen in the presence of oxygen. It will just not utilize oxygen. Just like light independent reactions, we call it dark reactions, but they can also happen when light is present. They can also happen during the day, but they are just light independent. Do you understand, Mariam, what I'm trying to say?
Yes, sir.
These are the basics. So I am asking, if there is an anaerobic bacteria, in the presence of oxygen, will it be able to respire or not?
It will.
It will.
Yes.
So what do we mean by anaerobic? So if oxygen is, so it should not, so there should be no effect of oxygen at all. Do you understand what I'm trying to say? There are two sentences. Listen to it. So earlier in AIMS, when AIMS used to have its own entrance, there will be a sentence which is assertion, and there will be another sentence which will be a reason for that. And then you have to tell that whether the reason is the correct reason for that assertion or not. So I am telling you one sentence, which is, there is a anaerobic respiration in which oxygen is not required. It can happen independent of oxygen. And there is an anaerobic respiration which can only happen in the absence of oxygen. What does these two sentences means?
One is obligate and the other is not.
Yes, what is obligate? What does the word obligation?
Without it, it cannot happen.
No,
I
with
mean,
it cannot happen, because we are talking about aerobes. So the difference is that if an organism is a facultative anaerobe, then the presence or absence of oxygen will not affect it. Even if oxygen is there, it can still continue to do respiration because oxygen is not required. So it's independent of oxygen. Okay. So facultative anaerobes can grow both in a oxygen positive and an oxygen negative environment. Is it clear? Environment. Is it clear, everyone? So facultative anaerobes can grow both in O2 positive and O2 negative environment because it has nothing to do with O2. Correct? If something is obligate anaerobic, it says that to be an obligate, there is an obligation on you. If there is oxygen present, you will die. So they can only grow in oxygen negative environment because oxygen for them is toxic. Do you understand the difference?
Yes, sir.
Similarly, there are facultative aerobes and obligate aerobes. Now, what does that mean? Facultative aerobes means they can grow like if there is oxygen present, they can use oxygen and become aerobic. And if there is no oxygen, then they will switch to some other mode, which is an anaerobic mode. You understand? But obligate aerobes means they can only survive in the presence of oxygen, which is us. Humans are obligate aerobic organisms. Is it clear, everyone? We
Yes.
have an obligation towards oxygen. If oxygen is not present, we will not survive. Our respiration will stop. So this is the broader outlook of respiration, aerobic and anaerobic. Is it clear? In plants, what are plants? Are they aerobes or anaerobes? They are aerobes. They are aerobic organisms. Are they facultative aerobes or obligate aerobes?
Facultative.
Okay, so if oxygen is not present, what else can they do?
Nothing.
Then how are they facultative?
But said they won't die in the presence of oxygen.
They won't die in the presence of oxygen. No, you don't have to. An anaerobe can die in the presence of oxygen. If you are aerobic, even if you are facultative aerobic or obligate aerobic, aerobic means that oxygen is good for you. You will never die when oxygen is present. You can only die if oxygen is not present. So plants, will they die if oxygen is not present or not? It's opposite of anaerobes. If oxygen is not present, will plants live or die?
Live.
How they will get energy?
Hi.
Mariam, how will they, what is the other way through which they can break down glucose? and
Yes,
do it?
they die.
They will die, right, Abhinav?
Yes.
Yes. So which means they are also obligate aerobes like us humans. And we are talking about aerobic respiration, therefore. In aerobic respiration, there is a process that happens which is called glycolysis. Now let's go to the terminology. What does glyco means? Glucose. And what does lysis means?
Breaking.
Breakdown. So we have to break glucose, glycolysis. And why are we breaking it? Because
Energy.
to produce energy. For the cellular and physiological processes to keep ourselves alive, basically. Now, how many of you have NCERT right now in front of you, or all of you have NCERT?
Yes, sir.
Yes. So can you go to the respiration part, and I'm sure that there must be a glycolysis pathway given. Can you open that pathway, Mariam?
Yes, sir.
Everyone keep that pathway in front of you. That is what I want, because what I'm going to tell you is that what's lacking in NCERT. So I don't have it right now with me. Okay, so can you see it? I urge, if someone does not have it, it's fine, because we are going to talk about it. But how many of you have it? Just tell me in front of you.
Sir, I do.
Okay, so let's start with this. In glycolysis, basically because we are breaking down glucose, it has to start with glucose, right? So let's start. I will write all the pathway with blue. Let's start with glucose. So we have glucose to begin with. And what is glucose? It's a 6-carbon molecule, correct? Let's write it out and keep doing it with me. So if you see, if Mariam, you can see, the first step of glycolysis requires use of ATP. Do you see it or not?
Yes, sir.
ATP, the book must be showing a thing like this, that ATP is coming in and ADP is going out. So it's utilizing energy, counterintuitive to what we were thinking. We were thinking that when we are breaking down glucose, energy should be released, but here energy is being used at the first step. So can anyone tell me why do you think it's happening? And we will also keep writing the enzymes. So this arrow, the enzyme that does this is called hexokinase or glucokinase, to be very specific. The word hexo means for 6 carbon, right?
Yes, sir.
Kinase means kinase means
Enzyme.
an enzyme, which basically what it is doing, phosphorylating, adding phosphorus. And remember macronutrients and micronutrients chapter for plants? There I told you what is the role of magnesium. It acts as a cofactor for glycolysis. So Mg2 plus as a cofactor. Okay, just write these information. I think this is given in your NCERT, the cofactors and things.
Sir, hexone is given. I mean hexane, but magnesium is not given.
So write down the cofactor because they are also asked. So, and glucose in the presence of hexokinase, or to be very specific, glucokinase, and the cofactor, this is a cofactor. If enzyme and the cofactor in the presence of which the enzyme works the best. So enzyme is hexokinase, cofactor is Mg2 plus. ATP is used, and what becomes? Glucose becomes glucose 6-phosphate. This phosphate goes, which came from ATP, goes and binds at the 6th carbon. It becomes glucose 6-phosphate. Is it clear, everyone? It is still a 6-carbon molecule, but now it has a phosphate attached to it. Now, why do you think it is happening? Why do you think at the very first step we are using energy for a process which was supposed to give us energy? Why in any biosynthetic or any biological pathway, whenever energy is committed, what happens? That step becomes, we have committed energy to this step. At the very,
Irreversible.
It becomes irreversible. Perfect, Mariam. So do you think that it is solving some purpose for the cell? It is deliberate, and this kind of pathway is evolved because it makes sense from the point of view of chemistry and biology. If at the very first step of the pathway, if you commit energy and make that step irreversible, then glucose 6-phosphate can never be converted back to glucose, right?
Yes, sir.
Which means glucose got into something. The identity of glucose inside the cell got lost, and now all the glucose becomes glucose 6-phosphate the moment glycolysis starts. Correct? Correct, everyone?
Yes, sir.
Which means more glucose which is waiting outside the cell can come inside, and there will never be a glucose saturation. You can keep bringing in glucose, and with the help of one ATP molecule, you will convert it into glucose 6-phosphate. Now it's dedicated. Now it is married to the glycolysis pathway and done. Its fate is written now. It cannot go back. So glucose glucose toxicity will never happen in the cell, because you will keep converting glucose into glucose 6-phosphate, and more glucose can come into the cell, right? So that is one benefit for biology. Okay.
Yes, sir.
So this phase, so what happens now? Glucose becomes glucose 6-phosphate. Now the second step is actually reversible step, because energy is not utilized here. So in this step, glucose 6-phosphate, in the presence of a molecule of an enzyme, again I'm writing all the enzymes here in black. So we convert glucose 6-phosphate into fructose 6-phosphate. What is fructose? What is fructose?
Isomer of glucose.
Perfect. And why are we doing this thing? Because again, just to seal the fate. But this is reversible. Fructose 6-phosphate can become glucose 6-phosphate, and this conversion can happen because it's reversible. We are not committing energy to this step, and it happens because glucose is being converted into its isomer. So can someone guess the name of the enzyme?
Isomerase.
Yes, ase should be there in the name of enzyme. So the enzyme should be isomerase, right? It's making isomerase, but it's making isomer of glucose. So it should be glucoisomerase. Clear?
Yes, sir.
And this glucose is a glucose phosphate. So it should be phosphoglucoisomerase. Just simple. Easy to remember. No mugging up. Phosphoglucoisomerase converts glucose 6-phosphate to fructose 6-phosphate. And it's a reversible reaction. Is it clear, everyone?
Yes, sir.
Still, it is a 6-carbon molecule. Okay. Now, at the next step, what happens? Again, let me use another color. Scheme should be again we are committing energy. Now, the moment we are committing energy, we are making it irreversible, correct? Now, why do we need to commit energy? Is because we have isomerized glucose 6-phosphate, made it fructose 6-phosphate. Now we want to add more phosphorus and make it fructose 1,6. Sorry, 1,6. Now there are 2 phosphates, it will become bisphosphate. Is it clear, everyone?
Yes, sir.
Still it is a 6-carbon molecule. We have not started breakdown till now. We have just utilized energy and made fructose 1,6 bisphosphate. Can you, now we have added phosphate. So can anyone guess the name of the enzyme?
Phosphates.
Kinase add phosphates. See above, hexokinase added a phosphate. So kinase should be there in the name. Is it clear, everyone?
Yes, sir.
And so this was a glucokinase or a hexokinase. This will be,
Fructokinase.
yes, fructokinase. And the fructose is already with a phosphate, so it will be phosphofructokinase. Clear, everyone?
Yes, sir.
And kinases needs a cofactor which is Mg2+. Nothing to mug up, just understand the story. Fructose 6-phosphate becomes fructose 1,6 bisphosphate now. Clear? The second step where we committed energy. Till now, still it's a 6-carbon molecule. Let me go little down. Now what happens? This fructose 1,6 bisphosphate undergoes a change where it becomes, what was the end product? Sorry, what was the first stable product when we were fixing CO2 in Kelvin cycle?
G3PH.
Sorry?
G3.
G3P.
Yeah, G3P.
Yes, that was glyceraldehyde 3-phosphate, correct? Glyceraldehyde 3-phosphate. Actually, we are converting it into a aldehyde. Do you see now? Fructose 1,6 bisphosphate becomes glyceraldehyde 3-phosphate. And we know that in a Kelvin cycle, this molecule was, how many carbons were there?
3.
3 carbon. So how many molecules will be made?
1.
2. 6-carbon molecule got broken down into a 3-carbon molecule. So there will be 2 molecules of glyceraldehyde 3-phosphate. Yes or no? Yes or no?
Yes,
Simple,
sir.
no? It's 6-carbon molecule got converted into glyceraldehyde 3-phosphate. So basically this is the first step where we started breaking down from 6-carbon to 3-carbon, which means how many molecules will be there?
2.
2. And it is a irreversible process because we have not committed any energy here. And we are converting it into a aldehyde. So what will be the enzyme known as? Anyone can guess? Ase should be always there, but the enzyme is called aldolase. To convert something into aldehyde, aldolases. Is it clear?
Yes, sir.
And it has a cofactor which is zinc, Zn2+, another element, micronutrient, zinc. So aldolase, zinc 2+ is the factor, and fructose 1,6 bisphosphate gets converted into glyceraldehyde 3-phosphate. Is it clear?
Yes, sir.
Till this step, we have started breaking down. So breakdown has now finally started. But till this step, there is no energy produced, only energy is utilized, right? Right or not?
Yes, sir.
Yes, sir.
Yes. So this step 1, step 2, step 3, and step 4. These first initial 4 steps of glycolysis are together 1 to 4 steps are together called preparatory phase. They're called preparatory phase, or in other words, they're also called energy investment phase. It's like a business. You have to make investments to get returns, right? So everything till now was spending energy. So this was preparatory phase or energy investment phase. Is it clear, everyone?
Yes, sir.
From this step onwards, we will be doing some, we will be making energy. In order to make energy, what is a good business like? A good business is where the amount of investment that you have done, you first recover that and then you get the profit, correct? Yes or no?
Yes, sir.
Cell here also is doing a business where it has to make more and more energy for its benefit. So these first 4 phases were very easy. They were easy because not much of change or the breakdown has just started. In the beginning, we were just converting it into so look here, you took glucose and you realize that if it stays in the form of glucose, it will not allow more glucose to come in because it will saturate. So you convert it into glucose 6-phosphate now by utilizing energy. So you sealed the fate. That's the first step of any good business. Then you just isomerize it from glucose 6-phosphate to fructose 6-phosphate, because now fructose 6-phosphate with the use of phosphofructokinase and energy can be converted into fructose 1,6-bisphosphate. Now you have 2 phosphate molecules on a 6-carbon chain, which makes it easy to break it. Give 1 phosphate to each 3-carbon chain and make 2 molecules of glyceraldehyde 3-phosphate. Is it clear, everyone?
Yes, sir.
So in case of step number 3, fructose 1,6-bisphosphate, 1 phosphate was on first carbon, other phosphate was on 6th carbon. So if you break it from the center, you can make glyceraldehyde 3-phosphate, right? Where both the molecules are basically having phosphate on the 3rd carbon because glyceraldehyde end is towards the center. Is it clear?
Yes, sir.
Yes,
The
sir.
first 4 steps are easy. Next steps are called payoff phase. So from this step onward, there are total 9 steps in glycolysis that you have to count. If they are 9, right? I remember it's 9, but I'll teach you is 9 step. Mariam, just count those steps in your NCERT. 9, right? Mariam, you're there. Mariam is writing. Yes, 9. Perfect. So 5th to 9th step is known as the payoff phase. I'm emphasizing on it more because a lot of question comes where they only mention payoff phase or preparatory phase in the question. So you don't get confused what part of the glycolysis cycle they are talking about. Or it is called energy generation phase. Energy generation phase. Yes, Mariam is writing. Oh shit, what happened? Sorry. Mariam, you're there. You're saying 10 steps. I think okay, I understood the 10th step you're talking about. Basically, there is a step after this where you are changing glyceraldehyde sideways into a dihydroxyacetone, right? Aldehyde is converted into a acetone. ketone. Aldehyde can easily be converted into ketone group, just chemistry. So that step I'm leaving for now. I'll incorporate that once I complete. Sorry, what happened? Once I complete this cycle. Is it clear, Mariam? Okay. Oh, we are out of time. Okay, so let's do one thing. The next steps are a little complex. I have to tell a lot of things about them. So shall we take it in the next class? I give you as a homework to go through the glycolysis pathway completely, all the steps, and try to understand it. And in the next class, we'll finish this pathway. Is it clear, everyone? Is that fine?
Yes, sir.
So let's take the payoff phase in the next class and go and revise whatever we have done today. Okay. So see you in the next class. If you have any doubts, please ask. Otherwise, I declare the class. Good night, everyone.
Thank you,
you,
sir.
sir.
Good night, sir.